Well, so far we've talked about epithelial tissues and we've talked about connective tissues. A third type of tissue we want to talk about in this video is muscle tissue. Okay, muscle tissue is not all the same. We'll talk a little about the differences between different types of muscle, and I think that that's and both how they function as well as how they appear. So let's go ahead and get started with that. First of all, a large part of my body weight is made up of muscle. Okay, at least 50% of my body weight comes from muscle mass. Okay, so there's a lot. It's a significant portion of my body. Though the term myology, myo means muscle, logi means the study of some muscle. Myology is just the study of muscle. And a couple things here that I think you should probably recognize is if you see a prefix sarco is a muscle prefix. It means muscle. Sarcolemma, where sarcolemma is basically the plasma membrane of a muscle cell, okay, which is actually more of a muscle myofibril or muscle fiber. Sarcoplasm is the sarcoplasm, the cytoplasm of a muscle cell. Sarcoplasmic reticulum, just like the endoplasmic reticulum that we see about in other cells. Sarcomere. Sarcomere is, I'll show you what that is shortly. It's the repeating structural units that are inside a muscle cell that are really required for contraction. And I'll show you what this means in a minute. And then we have a word like sarcoma. Oma means tumor. So sarcoma is basically a muscle fiber cell cancer. Okay, so we won't talk much about that, but what I really want you to recognize is if I see the word or the prefix sarco, it means muscle. It's a muscle prefix. Okay. Well, I mentioned that not all muscles the same. We have actually three different types of muscles that we should talk about. The first type is called skeletal muscle, and this is typical, the muscle that we usually think of more than anything else. Large body mass is the skeletal muscle. Second type of muscle we have is what's called cardiac muscle. Cardiac muscle is the heart muscle. Now, the heart muscle has some similarities to skeletal muscles, but it has some dissimilarities to skeletal muscle. Third type of muscle we have is what's called smooth muscle. Okay, smooth muscle is like muscle that lines the inside of my viscera or intestinal structures, vessels, smaller airways and stuff like that of the lungs. We'll talk about that. But anyway, if I look at these things, let me just go and try to describe these a little bit more. Skeletal muscles we call voluntary. And what I mean voluntary it means that I purposely cause those muscles to contract. If I want to raise my arm, my brain sends a signal down to the muscles of my arm, and my arm will raise. Now, I'm purposely able to direct that. So it's a voluntary type thing. I tell it what to do. Okay. They're what we call striated. Now, striated might not make much of a difference right now, but striated means striped. Okay. And this will be very clear when I start to show you some images of striated muscle. We know the skeletal muscle is also multinucleated. Muscle fibers or muscle cells are a little different from most other cells because basically what they are is they're really what are called myofibrils, these long strands, okay, we have one unit stacked upon another unit, stacked upon another unit, stacked upon another unit. And as compared to the typical cellular structure we have, it's a little bit different, okay? So therefore these long fibers or these long strands of muscle need to have multiple areas of control. So as a result, they become what they call multinucleated. That they're within a particular individual fiber and myofibril, there'll be multiple nuclei that are scattered throughout. They're highly vascular. And the reason why skeletal muscle is highly vascular is simple. Well, it's always working. If it's working, it needs a blood supply. It needs a blood supply for both nutrients as well as oxygen. So skeletal muscle is highly vascular. If you cut skeletal muscle, it bleeds like a fish, okay? Has a bony attachment for motion and for posture and muscle work. And what happens is the area where where the muscle becomes attached to the bone is called a tendon. A tendon attaches muscle to bone, and we saw that in our connective tissue video in regards to what that a dense connective tissue that we see with tendon looks like. Okay, so skeletal muscle is voluntary. It's striated or has a striped appearance, and I'll show you that in a little bit of cross striping. It's multinucleated because again, it's not the typical cellular structure that we normally see. We know it's highly vascular because it requires a lot of nutrients, oxygen to be able, because it's constantly working. Bony attachment is necessary, and that's by what we call a tendon. Now, cardiac muscle in some ways is similar to skeletal muscle. It's skeletal because it has the same striped appearance. And again, that'll become a little bit more apparent to you very shortly. However, cardiac muscle is involuntary. I really can't tell my heart to stop or to go faster or anything like that. It does what it wants to because of what we call the autonomic nervous system, which actually directs the rate, as well as, or I can't say directs the rate, because the rate of the heart muscle contraction is actually inherent in some special cells that we have in the heart that we'll talk about later in the semester in regards to how the heartbeat originates. But my autonomic nervous system can control it, make it go faster or slower, which I don't really have a whole lot of control over. The cells are uninucleate, okay, and they're a little bit different in their structure as compared to skeletal muscle because they branch, which skeletal muscle doesn't. It's a long individual strand where cardiac muscle branches off, okay. It's highly vascular, again, because guess what? Cardiac muscle is working from well before you're born to the time you die. So as a result, it needs a blood supply with nutrients as well as oxygen all the time. Again, it has this cellular branching with what are called intercalated discs. And I'll show you there what those look like. It's where one branch could actually meet with another branch of multiple different, or as the cardiac muscle branches off the different branches coming off the main trunk of the cardiac muscle, it could actually interconnect or interdigitate with a number of other cardiac muscle fibers. Okay, and basically this is a heart muscle. Okay. Smooth muscle is involuntary. In other words, I can't change the size of my vessels. I can't make my intestinal smooth muscle work faster or anything like that. So it's involuntary. It's totally under autonomic nervous system control. Okay, it's non-striated. It doesn't have a striped appearance. It's more spindle-shaped. It's very spindle-shaped type of a cell. Also has one nucleus, which is very centrally located in the cells. And again, it's for the viscera. When I mean viscera, if I mention the word viscera, I mean organs, internal organs. Okay, as well as for blood vessels, and as well as also the smaller airways that we find in the lungs. We'll talk about that a little bit later. And this is just a little sort of like a chart comparison that shows you the difference comparing the four major categories, whether it's voluntary or involuntary, what it looks like, striated or non-striated, nuclei, how many nucleus does it have, and its purpose. Okay, so you can actually go, and this is just a summary of everything we've talked about on the previous slide. Okay. Now, again, cardiac muscle, it has this striped appearance. If I look at it, and again, when we look at some microscopic pictures, you see these little stripes that look like they go across from side to side. Has a stripe, and there's a specific reason why it does. It looks very similar to like the stripes that we see in skeletal muscle, these long fibers. What happens, the difference is cardiac muscle branches off, has multiple branches, which allows it to communicate with a larger number of additional cardiac muscle fibers, which is very important. Okay, and then we have smooth muscle, again, which is more spindle shaped, like I mentioned before. Well, what are some of the muscle characteristics that we should think about? First of all, muscle is called muscle because it's able to contract. Okay, has contractility, and by this it means that what happens is the muscle, when it's stimulated, it shortens. Okay, and when it shortens, it produces this force. It's an amount of work that's produced to cause it to shorten, and as a result, that shortening causes motion. So contractility means it shortens, and it produces, by shortening, it actually produces motion that will occur. It's excitable. Okay, what I mean by excitable is that nerves will actually tell it what to do. In other words, if it's a skeletal muscle, which is voluntary, I have a certain area in my brain, which we'll talk about later on in the semester. It sends a signal out through a nerve that will go out the spinal cord down to where that muscle is, and actually that nerve will send that stimulation that will stimulate the sarcolemma, or the cell membrane around the muscle, that will cause that muscle to contract. It actually sets into work on a number of individual sequences or steps that will allow that muscle to contract. So it's excitable because it can be excitable by nerve stimulation. It has conductivity, which means that what happens is once that nerve gets down to the muscle, there's a little area, a little junction called neuromuscular junction, where the nerve approaches or reaches right the surface of the muscle. It sends an electrical signal along the surface of the muscle, the sarcolemma itself, and that's called an action potential. And this action potential is a change in the electrical activity on the outside, which actually is what stimulates the eventual muscle contraction to occur. So it's conductive by these action potentials, or an electrical current or electrical stimulus that goes along the surface of the muscle cell, the sarcolemma, that actually causes it to contract. We know that muscle is extensible. What happens is you can actually take muscle and stretch it to almost 50% greater than its resting length. It stretches out a lot, okay? This is important because some muscles are called agonists, and other muscles are called antagonists, okay? If I extend my knee, if I take my knee and extend my knee out, my quadriceps muscles, the ones in the front of the thigh, are contracting to bring my leg up straight with my thigh. Okay, they are called the agonist muscles. On the other hand, the hamstrings, or the muscles in the back of my thigh, I have to be stretched a little bit, because what happens is as I'm contracting the muscles in the front, the muscles in the back get stretched out a little bit, and they are called antagonistic muscles. So the muscle usually on the opposite side of the limb would be the antagonist, as compared to the agonist, which is the one that's producing the motion. Okay, so therefore these antagonist muscles need to be able to stretch a little bit. Okay, so when the agonist is moving, the antagonist needs to stretch, and and vice versa. We know that muscle has to be elastic, simply because when it stretches, it has to come back to its normal relaxed shape and form. So that's a little bit of the elasticity that we see with muscle. So contractility, able to shorten, excitable by nerve stimulation, conductivity, able to take that nerve stimulation and produce that electrical gradient change along the surface of the muscle. It's extensible, it can be stretched a little bit, and it's elastic. It comes back to its normal shape. And these are some very specific muscle characteristics that we should think about. Okay, now let's talk about skeletal muscle first of all, because again, that's the largest mass. Okay, it has a different cellular arrangement than what we've talked about thus far. In the past, we talked about a cell membrane and a nucleus and all these organelles and stuff like that. Well, skeletal muscle is different. Okay. If I look at this, this would be a muscle fiber here, okay? And this muscle fiber is actually a collection of a number of what are called myofibrils. Myofibrils. These are long cylindrical type arrangement of these of what causes the base, what's the basis of the muscle, okay? And within these myofibrils, okay, and again, the myofibril is held together by a sarcolemma, or a cell membrane. So if you look at this picture up in the top, you see the muscle fiber there, but inside there you see all these smaller little areas, the myofibrils. They're all sort of like, wound together. They're held by a membrane around the outside. Okay, we'll talk about that later on in regards to how this works, as well as inside we have all these little myofibrils now that are bound together in this muscle fiber. So muscle fiber is just a series, a number of these little myofibrils, which basically then can be divided up into what's called a sarcomere. Okay, and a sarcomere, okay, let me get rid of a lot of this drawing up in here. Okay, a sarcomere is basically a repeating unit that occurs within this muscle, or muscle myofibril. Okay, what happens is so here'd be one, there'd be another one here, there'd be another one over here, and one after another after another. If you look here, there'd be one here, one here, one here, and this myofibril is a series of these sarcomeres that are lined up one after each other. So if I have a long tube like this of that myofibril, there'd be a sarcomere here, a sarcomere here, 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 and they're lined one sarcomere on top of the other. Okay, now that's not what gives it the striped appearance, sort of. We'll talk about that in just a second. Okay, what happens is that's called a sarcomere. Okay, so these sarcomeres are lined up in a myofibril from end to end, just like we see up in here. Here's one sarcomere, here goes another sarcomere, there'll be another one here, and there'll be another one out here, and stuff like that. And again, these myofibrils by themselves, so I have a fiber. That fiber is a series of myofibrils. Well, inside those myofibrils are a series of what we call filaments. Okay, and these filaments are little strands. Okay, And when we look at this picture, let me get rid of all these lines here. When we look at this picture here, and we'll be talking a lot about this when we talk about the muscle individual. Inside the muscle myofibril itself are strands, and these strands are either thick strands or thin strands. We have what's called the myosin, which is a thick filament. If you look at this purple area right here, okay, the larger purple strands are called myosin. The smaller green strands are called actin, the thin filaments. Okay, what happens is all these myosin filaments are held together at the center of a sarcomere right here, which is basically it's called the M line, which is right here, which basically is where it's attached to the center of the sarcomere in the middle. Okay, these large thick fibers are these myosin fibers. At the end of the sarcomere is what's called the Z disc. Z disc is this area out here, and that's where these actin fibers attached to, okay, at the end. So basically what I'm going to have, let me just draw it down here. I'll show you. Here's my actin filaments. Okay, let's draw some thin actin filaments. Well, I didn't want to do it that way. Let's get rid of that. Okay, let's do a thin actin filament. Actin filament, actin filament, actin filament, actin filament. And over here's an actin filament, actin filament, actin filament, actin filament, and they're attached to the end of the sarcomere at what's called that Z disc. That Z disc. Okay. Well, in between them are these thicker. I hope this works out okay. Oh, no. These thicker filaments that fill in here like this. And these thicker filaments are my myosin. And the myosin is held together in the center at this M line, right in the middle. Okay? And so as a result, the way a muscle contracts is if you look at this individual image here, and this is just giving you a little idea, which we'll talk a lot more about later on the road. If you look in the image right here, there's a little bit of overlap here of the actin and myosin. The myosin, the thick purple, the actin, the thinner green. What happens is eventually with muscle contraction, these filaments that are sitting like this, okay, are now going to be forced to go like this, and they interdigitate more. So now that myosin is going to actually be moving. The myosin pulls the actin this way. The actin gets pulled this way. So as a result, the striped appearance that you see is where we have overlapping of these filaments, and that's what causes that striated appearance. And then with muscle contraction, the filaments that are normally only a little bit overlapping, they become overlapped more. What does that do? It brings one Z disc closer to the other Z disc, which then shortens the overall length of that sarcomere. The sarcomere gets shorter because now I have those filaments that'll cross over each other a little bit more. Okay. So anyway, that's sort of like a little bit of a preamble. We'll talk a lot about this more in a lot more detail down the road. But again, because of these actin filaments, which are the thin filaments, the little green ones, and the myosin, which are the thick filaments, which are the purple ones in the diagram, they cross over each other, and when they cross over, it gives a striped appearance, creating that striated appearance when we look at skeletal muscle, which is actually very, very, very readily apparent. It's easy to see. You don't need any kind of special glasses to be able to see those. Okay, so anyway, that's that. So let me sort of like summarize here. What happens is we have muscle fibers. These muscle fibers are actually a combination of a number of smaller fibers inside called myofilaments, okay, or myofibrils, okay. These myofibrils actually have a number of small things inside which are called filaments inside, which are basically these overlapping strands of actin and myosin. And when a muscle gets stimulated, those actin and myosin filaments will cross over each other, will interdigitate, which brings the sarcomere which is going from one Z disc to the other Z disc closer, which causes muscle contraction, and the muscle gets shorter. That's it in a nutshell, okay? It's a lot more involved with that in regards to all the processes that happen about what happens on the surface of the cell in the sarcolemma, what happens with calcium, and a bunch of other stuff. Sodium, calcium, potassium, all these things are going to be important when we talk about muscle contraction, but that's what you need to know right now. So anyway, that's why the muscle has that striped appearance, though, because the overlapping of these basic small myofilaments, or thin filaments, actin, thick filaments, myosin, because that overlapping gives that striped appearance of a muscle. Okay, so if I look at muscle here, and it's sort of like a distance, this is under a low power. If I look at low power, you see all these little strands. That's about it. You don't see a whole lot. What else do we see? What else do we see in here? Oh, obviously, see all that fat? There's a little fat. You see all the little fat cells in there? Fat cells? You should know. Oh, look at the fat in there, the adipose. Stuff that we saw on previous images, okay, or previous videos. Here's some more fat, and here's some more fat. So that's a little bit of medium power. However, when I start to pop this up into high power, I start to see this striate appearance. And that striate appearance is because of the overlapping of the actin and myosin in the myofibrils. Okay, hopefully that makes sense. And you should be able to see all those little stripes that are on there, and they go crosswise. Okay, it's not longitudinal stripes, but crosswise stripes. And that's what we see. We see what are called A bands, which are the dark bands, which are where the area that overlap. I bands, where there's not as much overlap. Okay. And that's what we see when we start to look at it under a higher power. This is something that I know in the lab, if we were in the lab on the microscope, you'd be able to see this, and that's very characteristic, okay? I see some capillaries there, which is small blood vessels and stuff like that, but that's that striated appearance that you see in skeletal muscle. This is, again, looking the same thing that we saw before. You can see that striped, again, the dark bands and the light bands, because that striation going across the muscle this way, because of the overlapping of those actin and myosin filaments. We see the same thing in here. We see a muscle fiber. Here's a muscle fiber, and here's a myofibril. Here's another myofibril. See the edge of another myofibril, and basically a clump of myofibrils. And this right here, the image on the far right, actually is taking the muscle. Instead of looking at it longitudinally, it cuts it across. And as a result, we're looking at a cross section. So if I have a muscle, let me just do it. If I have a muscle here, this image in here and this image is looking at it this way. This image right here is looking at it this way. So it's cut across. So all the little dots that you see in there, those are all the little myofibrils and the filaments that are at the very end. You're looking at it sort of like at the end of that little filament. That's what you're seeing there. Okay, so that's what you see with the skeletal muscle. And again, but that striated appearance you see longitudinally is exceptional appearance. This is again looking at a skeletal muscle. You can see that striated appearance basically is because of that overlapping of the actin and myosin filaments that gives that strong striated cross-hatched appearance of skeletal muscle. Unmistakable. It's easy to see, and you see here's see the same thing. Striated appearance. Very classic. No branching, straight filaments. Just a long core of fibers. Okay. Now cardiac muscle again looks like skeletal muscle because it's striated. Okay, both of them are striated. So cardiac muscle is also striated. Does have that striped appearance, and because of overlapping filaments and stuff like that. The difference is where skeletal muscle goes like this, one sarcomere line on top and another sarcomere on top of the other sarcomere, cardiac muscle actually branches at the end, which allows it to meet with other branches of other cardiac muscles down the road, okay, our muscle cells down the road. So it branches at the end of the fiber, okay. So you can see right here how things are branching off. So there's a branching off, there's a branching off, and that's what we see. What happens is where that end of that cardiac muscle fiber ends, okay, it gets to what's called an intercalated disc, which actually is something on the microscope is very apparent, okay. So instead of having just cardiac muscle fibers just end to end, it branches off, which allows it to interdigitate with more cardiac muscle fibers, okay. Where skeletal muscle was multinucleated, basically cardiac muscle is a single central nucleus. Sometimes there might be two, but mostly I would say it's uninucleate with only one nucleus. Okay. Now, if we look at here, you can actually see you don't have quite that parallel arrangement that you saw when you looked at skeletal muscle. It branches off. Okay. And if I'm looking here at this, see these little lines right there? The little black line, 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 little black line. Those are the intercalated discs. And you can see how the muscle, cardiac muscle branches off. So it's able to meet with other more cardiac muscle. Okay, muscle fibers. So that's the difference between that. We still look and we still see that striated appearance of cardiac muscle. See that cross striation? You still see that cardiac muscle striation occurrence, but the difference is instead of having a bunch of parallel fibers, these fibers start to branch off at the end. Okay, they'll branch off at the end, and these branches allow those little connections are called those intercalated discs, where one meets another cardiac muscle cell. Okay, see the same thing over here. There's one, there's one, there's one, there's one, there's one. There's one, there's one. Those are all intercalated discs where they actually join the end of another cardiac muscle cell. Okay, same thing here. Okay, you see how it has more of a branching appearance. There's an intercalated disc, intercalated disc, intercalated disc, intercalated disc, intercalated disc, intercalated disc, intercalated disc. That differentiates it. If you see something that has it's striated, but it has all these little crossed areas like that are darker, which are those intercalated discs, you know it has to be cardiac muscle. Okay, so it looks that striated appearance just like skeletal muscle, but cardiac muscle branches, and where it branches and meets another muscle fiber, it's called an intercalated disc. Smooth muscle is totally different. Smooth muscle is involuntary, as we talked about. It's spindle shaped. Remember, spindle shaped? It's like that. It has a central nucleus. It's inside that. Looks like a little eyeball, I guess, maybe. Sits inside like that. Centrally placed nucleus, spindle shaped, and it is not striated. It doesn't have that same cross hatching of actin and myosin. So it looks a little bit more amorphous. You don't see that. The striations occurring in smooth muscle. This is smooth muscle. Under low power, it looks not that much different. You know, you see a lot of strands, but when you start to get a higher power, you just don't see any cross striations, any striated appearance of smooth muscle. It's basically a bunch of spindle-shaped muscle next to another spindle-shaped muscle, which is next to another spindle-shaped muscle, which is next to another spindle-shaped muscle, like that, and so on and so forth. And that's what we see, okay? And that's what we see with smooth muscle. Again, we see this lining the inside of blood vessels, allow vessels to actually change in diameter. Some of the smaller airways, the bronchioles, actually have smooth muscle. They're able to change in diameter to let more or less air in. When you're sitting there trying to be quiet and your stomach is sound like a bunch of Greek geese flying over, it's basically fluid material being moved through your intestines by a process called peristalsis, and that's because the walls of the intestine are squeezing to moving things down, sort of like milking all that material down through the intestines. Okay, and that's called smooth muscle. Here's more smooth muscle, and again, yeah, these are all nuclei that you see there, and they're basically in spindle-shaped cells. You know, spindle-shaped cells. That's what you see there. So that's smooth muscle. Nothing really exciting. It doesn't have that nice striped appearance like we saw with skeletal muscle, but that's smooth muscle. So what we talked about here today, we talked about muscle. We talked about some of the characteristics of muscle. We talked about some of the differences between muscle, both microscopically as well as functionally. We talked that skeletal muscle is voluntary, where cardiac muscle and smooth muscle is involuntary. It's controlled by centers in the brain, which I don't really have any control over in a thought process. I can't make my intestines work faster. I can't change the size of my blood vessels by thinking it. I can't make my heart go faster or slower just by thinking about it. The body has certain areas in the brain, which we'll talk about in the neurological system, that will help to control that, and that's called autonomic. Okay. On the other hand, skeletal muscle is voluntary, which means I could do whatever I want. Whatever I want to do, I could do. Whatever I want, all those muscles could work. Okay. They look bad. Okay, but anyway, I can make the muscles do whatever it wants to do. We know that what happens is when I talk about the nucleus, skeletal muscle is multinucleated because what? I have one sarcomere, which is basically a muscle unit, cellular unit, on top of another, or next to another, lined up in long strands. So therefore, I need to have multiple points of control, like different stations along a railroad to be able to control things. Where cardiac muscle and smooth muscle are uninucleate. Okay, we know that skeletal muscle and cardiac muscle are striated because of that actin and myosin overlapping filaments that we'll see that allow them to shorten. Smooth muscle is non-striated because it has a totally different system as to how it works. Okay, so hopefully by looking at the pictures that we showed, the microscopic pictures, you can see the difference between those. We know that a cardiac muscle is branched, where skeletal muscle is not. Okay, it's one sarcomere lined up next to another one. Okay, so that just tells you a little about the difference between the types of muscle. Hopefully this makes sense to you. Hopefully you'll be able to look at an image and pick it out as to what's what. And if you have any questions, like in everything else, you could go ahead and either email me or ask, or whatever the case may be. And other than that, be safe and be healthy. Okay, bye-bye.